Hi everyone, tonight's video is on DNA replication. So in front of you, you can see an unreplicated chromosome that has one chromatid. And during the process of DNA replication, this unreplicated chromosome becomes a replicated chromosome with two chromatids that are attached at the centromere. We can look at this in a slightly more detailed fashion, showing the actual base pairs on the left-hand side here. With the top half of this chromosome, it's in the process of being replicated. So the top half has one chromatid and the bottom half has two chromatids. Now, because you can see these base pairs, if you know the base pairing rules, it's not hard to imagine how a cell could copy its DNA. You can see this region of DNA right here where the arrow is has not been copied yet. Where there is a G, the cell would put in a C, and where there's a C, the cell would put in a G. So that seems pretty simple, but in reality, it's a very complicated process. So the first thing we have to do is separate the two strands of DNA. Remember, DNA is a double-stranded molecule, and the strands are arranged in what we refer to as an anti-parallel orientation. The strands each have a 5' and a 3' end. And if you look at these two strands of DNA in this DNA molecule, the top strand is oriented from the 5 to the 3' direction, going left to right, and the bottom strand is oriented from the 3 to 5' direction, going left to right. So this is an anti-parallel double-stranded piece of DNA. So to separate the two strands of DNA, this occurs at what we refer to as the origin of replication. This is where replication starts. And it's in the middle of the DNA strand, not the end. And I'll talk a little bit at the end of this video why cells need to start in the middle of the DNA strand. So the enzyme responsible for separating these two strands of DNA by breaking the hydrogen bonds between the base pairs is called helicase. Remember, these two strands of DNA are held together by hydrogen bonds between the nitrogenous bases of the individual nucleotides in the separate strands of DNA. As I said, when these strands separate by the enzyme helicase, it forms what we call a replication bubble. And we can actually see this in the electron microscope. If you look at this picture here, you can see two strands of DNA that have been separated by the enzyme helicase, forming a replication bubble. So beginning a new strand of DNA is very complicated, and that's because DNA polymerase, the enzyme responsible for making new DNA, has some sort of quirky characteristics that make it hard to work with. The first is that DNA polymerase cannot start making a new DNA strand by just putting together two nucleotides. In science speak, we refer to this as it cannot start de novo. DNA polymerase can only add to the 3' end of an existing strand. And my students every year find this so confusing. Like, why can't it just put two nucleotides together if it can add on to a 3' end? Well, here's what I usually explain it as. To put two nucleotides together, you need one nucleotide in each hand. So you'd need two hands to put them together. You can think of DNA polymerase as only having one hand. With one hand, you could add a nucleotide onto an existing strand, but you couldn't put two together because you couldn't hold two in one hand. So I hope that visual helps you understand that. So to get around this problem, cells use an enzyme called primase. Primase is a special form of RNA polymerase that can start de novo. You can think of primase as having two hands. It can put two RNA nucleotides together and then continue building those on the 3' end of the growing primer. Both RNA and DNA can only be extended on their 3' ends. You cannot add on to the 5' end of a DNA strand or an RNA strand. So primase, our RNA polymerase, starts making a primer starting at the origin of replication, and it uses the base pairing rules. It's reading the bases in the DNA and putting in the correct base, but it's using RNA nucleotides. Of course, it has to put in a U instead of a T when it sees an A in the DNA because RNA doesn't have T, it has U. So primase is going to make a short RNA primer starting from each origin of replication, and that's going to give DNA polymerase something to add on to. Once the primer has formed, DNA polymerase, and if you're in an advanced class, that would be polymerase 3. If you're in an introductory class, you don't need to know the individual polymerases. But DNA polymerase would add nucleotides onto the 3' end of each primer using those base pairing rules. Now, the hardest part about this is knowing which direction to go from the origin of replication. And you might be wondering, how did I know to start drawing my primer going from the top origin of replication going to the left-hand side, and from the bottom origin of replication going to the right-hand side? Well, I know two things. First of all, that the two strands of DNA are always anti-parallel. So the new strand of DNA has to be built with the growing 3' end going towards the 5' end of the existing strand. So if we see our new strand of DNA, this is its growing end. This is its 3' end. If it kept on growing here, it would eventually be opposite the 5' end of the existing strand, giving it the anti-parallel conformation. I also know that you can only add on to a 3' end. So with those two pieces of information, you can always figure out which direction to go. Again, on the bottom strand, my growing end has the 3' end, and if I kept extending it here, that would be up against the 5' end of the existing template DNA. So now we have another problem. I told you that DNA and RNA polymerases can only go in one direction. They can only add on to the 3' end of both RNA and DNA. So how does the cell copy sort of behind the origin to the 5' end of the origin? Well, what the cell does is primase simply starts a new primer going in, again, that 5' to 3' direction, and it would start a new primer here. Then that gives DNA polymerase, again, polymerase 3 if you're in an advanced class, something to extend onto so we can extend our DNA strand. So the replication bubble keeps growing in both directions. It gets bigger and bigger, and as it gets bigger on this end, the cell has to start with a new primer and adding on to more DNA. If we looked at this replication bubble and divided it in half, we say it has two replication forks. I mean, it kind of looks like a fork if you look at it. Let's look at the left-hand fork, the one I have shaded in orange here. On this top strand, the DNA strand can simply keep growing on its 3' end as this replication bubble opens. As these strands separate, this 3' end is just going to keep on going. We call this the leading strand because as the replication bubble opens, it keeps on going. It doesn't have to start and stop. If you look at the bottom strand, as the replication bubble opens, the new strands have to be started with a new primer and then DNA polymerase coming on and adding on to our primer. So it's made in segments. We call this the lagging strand, and it has to do with time. The leading strand it's faster, so it leads the process, and the lagging strand takes longer because of the more enzymes involved in the process, so it lags behind. We can look at the right-hand fork of my replication bubble and see the same thing, only in this time the leading strand is on the bottom strand, because this is the one going in the 3' direction to end up opposite to the 5'. Whereas the top strand will now be our lagging strand that has to be made in fragments with the primer starting and then another extension by DNA polymerase. At each replication fork, we have a leading strand that's copied continuously and a lagging strands that are copied in fragments. We call these fragments Okazaki fragments. That's a very important term. So the fragments that are made with a primer and a DNA extension on the lagging strand are called Okazaki fragments. So now if the replication bubble has moved open to the point where the strands are completely separated and we've finished copying our DNA, we have two problems with our new DNA strands that have been made up against our old DNA strands. First of all, there's RNA in my new DNA strand. Remember, primase makes an RNA primer. Those primers get removed and filled in with DNA by another enzyme called DNA polymerase 1. You only need to know that if you're in an advanced class. Otherwise, simply DNA polymerase would be fine. So these primers get filled in with DNA. So that's great. We got rid of our RNA in the DNA. But we have another problem in that our DNA is in pieces. So the lagging strand that was made in small pieces, that DNA at the end of the 3' end can't join that 5' end. We're going to need another enzyme to kind of glue these Okazaki fragments together. And that enzyme is called ligase. And ligase forms phosphodiester bonds, which are a type of a covalent bond between these fragments, so that we now have 2 cohesive strands of DNA in each of our new molecules of DNA. If we look at these new molecules of DNA, let's go back to the picture I showed you at the beginning of this video, where the top half still has 1 chromatid and the bottom half of the chromosome has already been replicated. You can see that each of the new molecules of DNA has 1 old strand from the original molecule and 1 new strand. We call this semiconservative replication because in each of the new molecules of DNA, 1 of the old pieces of DNA has been conserved. And I also promised you at the beginning of this video to talk about why origins of replication are in the middle of a DNA strand. Well, the answer is that cells have millions of copies of base pairs to copy, so they have to use multiple origins of replication, each of them using all of the enzymes we talked about today at the same time. This is kind of multitasking. If the cell just started at one end and worked its way to the other, it would never finish the process. But by operating in multiple origins of replication, the process can be copied in much less time. And you can see here an electron microscope where we have 1 strand of DNA with multiple replication bubbles open at the same time, the result of multiple origins of replication. So today, after this video, you should know you should be able to draw DNA replication, including the roles of helicase, primase, DNA polymerase, and if you're in an advanced class, you should know the difference between the roles of DNA polymerase 1 and DNA polymerase 3. If you're in an introductory class, just DNA polymerase is fine. You should know what an Okazaki fragment is. You should know what the role of ligase is and how that relates to our Okazaki fragments. And you should always, always, always be careful of your 5' and 3' directions. And finally, if you are in an advanced class, you should probably know the roles of topoisomerase and single-stranded binding proteins. But again, these are not necessary if you're in an introductory class. So that's all for tonight.